I'm grateful for the opportunity as part of this symposium to present work on editing of CF patient-derived stem cells for functional restoration of CFTR in airway epithelium. And I have no relationships to disclose. So I'd like to start with a consideration of how we can apply gene editing for the treatment of CF. And the work here is really going to be focused on the 7 to 8% perhaps of individuals with CF who are not producing sufficient protein that can be acted upon by modulators for restoration of CFTR function. So we're talking primarily about genotypes that are stop codons or splicing mutations or perhaps significant deletions, for example. And the hope is that once these editing strategies are utilized successfully in such individuals, that they may be more widely available for a lifelong cure. So the one paradigm I'd like to sort of explore is the one that's shown here, that starting with, and I'm going to focus primarily on the airway, that if one obtains sort of patient-specific airway epithelial cells and has a way then of identifying and expanding stem cell populations, would be then ex vivo to correct the CFTR mutation in such cells, and then develop ways to successfully transplant these corrected cells back into the same patient's airway for an autologous cellular therapy. And the focus here on stem cells is essentially for a long-lived or perhaps lifelong benefit. One can also imagine, rather than using directly the primary airway stem cells, is to otherwise generate induced pluripotent stem cells, iPSCs, from the individual, correct those iPSCs, and then similarly derive these airway stem cells that can be used in transplantation. And while we're exploring these methods of optimizing the gene editing and correction in the stem cells, the hope is that there may be ways of appropriately using vectors or delivery tools to introduce these editing reagents directly in vivo into the patient's airways for correction in vivo in a way that doesn't require either transplantation or subsequent regeneration after the introduction of those cells. So I'd like to sort of talk about what type of stem cell population that we're most interested in, and that is really this airway basal cell. And what's been demonstrated now is that in a number of studies that following injury, that these basal cells have the ability to both self-renew as well as through differentiation to give rise to the variety of luminal cell types that appear in the airway. So this is just showing the pseudostratified proximal airway epithelium with the basolateral located basal cells, and then more luminally exposed the secretory, ciliated, and ionocytes. So the idea is if we can somehow successfully edit the basal cells, that we may be able to give rise to progeny cells that are likewise corrected and express CFTR at the appropriate level. So here's the overall paradigm that I'm going to be primarily discussing today, is starting with these primary airway epithelial cells obtained, for example, by a brushing, then a selective expansion of the airway basal cells, either in a feeder-independent or a feeder-dependent methodology, then to perform the editing, and then one would have a subsequent pool of cells, some of which are correct, some of which are uncorrected, which could then, if the efficiency was high enough already in this pool, could be expanded and then used for transplantation perhaps, or could be single cell cloned to derive uniform populations of cells, all of which would be corrected for the CFTR mutation. So the idea is we want to convert essentially a mutant CF airway epithelium to sort of normal function by, for example, taking cells from the airway, ex vivo editing them and returning them back in sufficient frequency to give rise to corrected cells that could then be hopefully lifelong regenerated by the presence of these corrected airway basal cells. So I'm going to talk today about two overall strategies for editing the endogenous CFTR locus. The first is one of site-specific correction or mutant-specific correction, where one would take, for example, I'm just going to use the example of F508 del here. One could develop specific sort of reagents targeting the mutant sequences, in this case by zinc finger nucleases. It can also be done by CRISPR-Cas9. We've done that as well. And then to introduce double-stranded DNA break, and then the introduction of wild-type donor sequences in various form that can then recombine to give rise to the corrected sequences. There are now other methodologies developed that don't actually require the induction of a double-stranded DNA break, be it base editing or prime editing. So these are also very, very promising approaches. So what I'd like to do is just show you briefly some work that we've published on homozygous F508 del airway basal cells that were first electroporated with zinc finger nuclease messenger RNA. This ZFN was specifically targeted to the mutant sequences, and then donor sequences were delivered either by electroporation of single-stranded oligos or delivery of AAV vectors of a larger homology sequence. And then the desired end product would be the corrected, that is the replacement of the mutant sequences with the wild type. One can also have alleles that are completely unmodified by this approach, or simply indels that were introduced by the nuclease. So shown over here, I'm just going to focus on the AAV delivery. That you can see here is in a bulk population of cells, we have 31% of alleles that are actually corrected. We have 49% of alleles which are unmodified, and about 20% that retain indels. And of these corrected alleles, there may be some cells that have both alleles that are corrected, some cells that only have one allele that's correct. This is just measuring at a bulk allele percentage. And what we have shown is that when we take those bulk population of cells, put them into Allele culture, that they give rise to well-differentiated epithelium. We show restoration of CFTR protein, in this case band C. And then when these cultures are subjected to Ussing analysis for CFTR specific current, you can see that we have levels of restored activity, in this case approximately 40% of non-CF, which is a significant level in itself for a level of modification that's somewhere in the sort of the high 20s to mid 30s efficiency of the actual correction. In the case of the single-stranded oligos, we have lower levels in our hands of both the genome modification as well as lower levels of CFTR current. So this is definitely an approach that can be pursued, and Matt Porteous' group has also showed by CRISPR-Cas9 the ability to efficiently correct with these methods as well. Now, what we focused more of our effort is really more on a universal approach shown here, because CF can result from such a variety of causative mutations. We would like to develop methodologies that, in fact, can use one or small number of tools to, in fact, correct for all genotypes. And in this case, what we've done is begun exploring at different introns in the CFTR locus, the ability to introduce double-stranded DNA breaks, in this case by zinc finger nucleases. We've done it as well for CRISPR-Cas9. And then to deliver the donor, again by AAV, in this case with the exons 9 through 27 as a cDNA, in this case codon optimized, preceded by splice acceptor, and followed by a polydenylation site. So the idea is that one has, once successfully targeted, one has the normal transcription coming from the promoter here and proceeding down through the polydenylation sequence, and then with the appropriate splicing, the goal is to end up with a wild type CFTR messenger RNA and protein. And this proved in our hands to be very, very efficient. That, for example, shown here is that we have 56% of alleles shown to be targeted in the desired way. And this was a sufficient frequency that we could actually pick it up by southern blotting here for the presence of the incorporation of the CFTR half gene in this case. We showed that bulk populations of these treated cells, again, were unaffected in their ability to generate air-liquid interface cultures. And we showed restoration of CFTR protein. And importantly, then, when these cells were analyzed by Biopsy chamber analysis, we saw between 34 and 42% of non-CF activity in these bulk populations of treated cells. And those data points are shown over here for the levels of genome modification approaching 50% or so. And here are the levels of the non-CF current that were observed. So we wanted to see what was really happening with these bulk cultures, and in fact, to see whether we were observing the desired splicing specifically into the transgene sequences. And what's shown here is that this is our desired targeting and desired splicing, so that all of the messenger RNA would now be of the corrected form. But we consider that maybe there could also be splicing across the transgene that would basically just give rise to the endogenous mutant messenger RNA and defective protein, if you will. So what we did is we developed, in this case, 4 single-cell derived clones. All of them proved to be homozygous for targeting into intron 8. And then we looked at the relative percentage from those clones of messenger RNA, either that was appropriately spliced into the transgene, here between 58% and perhaps 90%. But we did see, depending upon the clone, some level of message that really reflected simply splicing across the transgenic sequences, giving rise to the mutant. So this may help explain why that level of restored CFTR function was not quite as high as we were targeting. But there are things we can do to optimize, if you will, the specific splicing into the transgene sequences. So this was all done in these F508 DEL lines that were successfully targeted. And together with Ann Harris and her group at Case Western, what we wanted to evaluate is whether the introduction at high frequency of these cDNA sequences into intron 8, whether it in fact disrupted at all the pattern of open chromatin that is characteristic of airway basal cells. We wanted to make sure that we were not going to either disrupt the open chromatin configuration or somehow really affect that in such a way that would result in not getting the correctly cell type specific and levels of expression that we were hoping for. And in fact, we showed here that whether we look at the cis-regulatory elements or the promoter, for example, we see minimal effect of our targeting on the appearance of open chromatin. We do see a slight increase in open chromatin at the position of our cDNA. We don't know the reason for that yet. So this was all done in the homozygous F508 del. We were, as I mentioned initially, primarily interested in applying this now to genotypes which are not susceptible to modulation. So for example, in this line here, which is a stop-stop, we see 62% of targeting. We confirm that again by southern blotting. And importantly, when these cells are put into allele culture, we see restoration of, for example, good levels of non-CF CFTR channel activity. So we believe we have a methodology here that's quite useful, and we're now working on sort of moving this targeting into different introns. I have data in the one slide from intron 7. We're now moving further up, all dependent upon our ability to properly vector these donor molecules. We've shown that these basal cells targeted with the TI8 are in fact able to engraft in this tracheal xenograft model, so that's encouraging with respect to eventual transplantation. So we now have this pool of cells that is a mixture of corrected and uncorrected, and we wanted to really develop methods that could expand these cells either as a pool for subsequent transplantation or perhaps to develop single cell derived clones. And this just shows a selection of a media with supplements that allows us to get robust expansion, maintenance of basal cell specific markers, and most importantly, when placed in allele culture, robust maintenance of CFTR activity even after multiple passages and formation of multipotential airway epithelium. In the single cell derived clones, we in fact found quite a variation in the level of expansion, multipotential differentiation, and CFTR function. And you can see some clones here that behave beautifully in terms of differentiation, thick air-liquid interface culture, as well as robust CFTR function. And you see others here that are more heterogeneous in the level of differentiation, lower level. So our thought is that we would in fact select for those clones that demonstrated this multipotential differentiation and CFTR function, and those would be the ones that could potentially be used for therapeutic purposes. Now, I focused all of my discussion here on these primary airway basal cells, but in fact, one can imagine also starting with iPSCs derived from the same patients, performing the correction, and then deriving specifically airway basal cells from these iPSCs, clonal populations uniformly corrected, with the idea of using those perhaps in transplantation studies. And in another symposium, I presented a talk focusing mainly on this issue of derivation of airway basal cells. So I'd like to direct your attention there if that is of interest to you. So in conclusion, we now have a variety of editing methodologies capable of correcting CFTR mutations directly either in primary airway basal cells or an iPSCs that can be from which airway basal cells can be generated. And we're now continuing to sort of optimize these methodologies. Of course, we've looked at off-target effects in many of these, and I'll direct you to our molecular therapy paper if that's of interest for you with respect to the zinc finger nucleases. I'd like to thank the people in my laboratory, as well as the valuable collaborations we've had, all of which have played a key role in taking some of these methodologies forward.
Hello, everyone. Today, I'm going to describe the work that we have been doing to characterize the epithelium of the airways in cystic fibrosis. In particular, I will focus on the description of basal cell, they are the stem cell in the airways, and on the transitional state that we found to be modified in the disease context. I don't have anything to disclose in relationship with this presentation. We know that cystic fibrosis is a genetic disease, and is caused by mutation on the Cftr gene. Cftr is a protein that is important to maintain a proper composition of the mucus of the airways. When Cftr is not functional or is not expressed, we assist to a thickening of the mucus. There is a phenotype that is typical in cystic fibrosis. Here on the left are all the steps going from DNA to a functional protein. And during the year, each one of these steps have been targeted for therapy with different level of success. In particular, the last two steps, the one related to the misfolding of the protein and to the amount of chloride that can be transported from the protein, are the one in which therapies have been more successful. Today, we have drugs that can be used also in combination that target these two aspects of the production of Cftr. And these allow to really ameliorate life for majority of patients with Cftr. Unfortunately, there is a minority of patients in which these modulators cannot be effective. And for this percentage of people that it is important to pursue the development of gene editing that allow to introduce a DNA that will produce a functional protein. And to do that, we really need to understand what is the cellular composition of the epithelial cell that line the airways in cystic fibrosis. What are the characteristics of the subset? What are the differences in CF compared to control? In this cartoon, we visualize a situation in which in blue, we are able to introduce a correct form of the gene that codified for Cftr. And this ideally will lead to a situation in which the mucus composition can be reconstituted and therefore the functionality of the airways. This information and this concept is really behind our idea to undertaking the analysis of the single cell level of the transcriptome in cystic fibrosis. This was done by a consortium put together by the Cystic Fibrosis Foundation that include 3 different labs. Bridget Gompert at UCLA, as at the Cedars-Sinai Medical Center, and the Cystic Fibrosis Foundation lab. We were able to collect a large amount of sample, I'll show it here in this cartoon, from different portions of the airways. And altogether, we were able to collect 19 control and 19 cystic fibrosis samples. One big advantage of having this big collaborative effort was that we were able to analyze our data and focus on those observations that were maintained between the different groups, adding a level of robustness to our analysis that really was very important, because now we can use the atlas that we produce, summarized here in the center, and interrogate all the major cell type, basal, secretory, ciliate, and other smaller specific cell type, as for example, one for the cell, ionocyte, and neuroendocrine cells. But in particular, we can really dissect characteristics of subset of basal, secretory, and ciliated cell, and find their functionality and what is changed in the context of cystic fibrosis. For example, here on the right is an image that shows ionocytes. There are specialized cell types for the production of Cftr that have been recently discovered. And we can go back to our transcriptomic atlas and see where Cftr is expressed, not only ionocyte, but in general in the epithelial cell in the lung in controlling CF. Here on the left is the percentage of Cftr positive cell in controlling cystic fibrosis. And you can see that we do see ionocyte present, but we can also see that subset of secretory cells as well of basal cell contribute to the expression of Cftr. And on the right here, if we look at the level of expression, we can notice that those same cells that do express Cftr tend to have an increased expression in CF compared to control. And this is true for ionocyte, secretory subset, and basal subset, indicating an attempt of this cell in CF to increase the production of cystic of the CFTR itself. In a parallel experiment, a project that we perform in collaboration with the Big Boucher group and Okuda Kenichi at UNC, we asked the question if there is any difference in CFTR expression between large and small airways. And what we were able to show is that indeed also in the small airways, ionocytes do contribute to the production of CFTR, as well as secretory and basal cell subsets. Overall, this gives us a picture in which the airways of the lung contain this rare and specialized cell ionocyte for the production of CFTR, but also subset of secretory cell and of basal cell contribute to the overall production of CFTR in the lung. And this is important. In particular, it's interesting the fact that subset of basal cell may be contributing, because we know the basal cell are the stem cell in the lung that can themselves differentiate in secretory as well into ionocytes. And so for us, it was really important to dissect in more detail the diversity of basal cell. And here is a summary of the 5 different subsets of basal cell that we are able to identify. And what I want to do now is go through some of the transitional states that we identified in these different subsets and how these diverge in control compared to cystic fibrosis. Here is an example of the analysis that we did to characterize the proliferative capacity of basal cell in control in cystic fibrosis. These are a section from control lung and cystic fibrosis airways. And we are looking here at the immunoreactivity for the keratin 5, there is a marker for basal cell in green. The PCNA, there is a marker for cyclin cell in red. We can appreciate how in cystic fibrosis, there is a reduction of the colocalization of the 2 markers, indicating that there are less basal cell proliferating. And this is maintained true also if we harvest cells from both control and CF. If we culture those basal cells as primary culture, also in this context, we do see a reduction of proliferation in CF basal cell compared to control. Nevertheless, if we extend the culture for a longer period of time, what we observe is that the basal cell from CF rescue their ability to proliferate in a similar way to what we observe in control lung. This is an important observation because it does indicate that the impairment in the proliferation that we see in CF basal cell is not related to an intrinsic problem in basal cell itself, but is most likely related to the microenvironment in which basal cell are located. And this is an important consideration when we want to consider using a specific subset of basal cell for cell-based therapy. Here on the left is a so-called dimensional reduction in which each dot represents a cell, and the position of each dot is in relationship of how similar the transcriptome of each cell is between each other. And it is color-coded to describe basal cell in green, secretory cell in orange, red, and ciliated cell in blue. Here, if we focus on this branch of the graph, we can see here this basal 4 subset of basal cell and this ciliated 1 subset of ciliated cell. And we found that in cystic fibrosis, this specific subset of basal cell have the tendency to start expressing genes that are related to early transition to ciliated cells. And here on the right, we perform an in situ hybridization to label RNA from control and cystic fibrosis airways. And in green, we label keratin 5 for basal cell. And you can see here basal cell in parabasal position above the layer of basal cell. That is also co-labeled for LRRC6. There is a marker of early ceogenesis. And this indicates that in cystic fibrosis, there is an attempt of basal cell to directly differentiate into ciliated cell, while in the healthy lung, we know the basal cell transition to a secretory cell, and from there, there is differentiation to ciliated cells. If we look at this other branch between this subset of basal cell B3 that we call secretory prime basal cell and this specific subset of secretory cell, we identified this other subset of basal cell. They are primed to become secretory. Here on the right, we see immunoreactivity for characterize the labeled basal cell. In association with CD66, there is a marker that we specifically identified to be associated with this secretory prime basal cell. And here we can see some basal cells that do co-stain with the two markers, indicating that they are cells that we can find in the human airways. The information that we were able to collect about the diversity of basal cell was also very useful for us to be able to really fractionate a different subset of basal cell and perform in vitro experiment and better study their characteristics. Here is an example of surface marker that discriminate for this subset of basal cell, and we were able to use specific fractionation by flow cytometry using specific marker to compare subset of basal cell from freshly isolated lung compared to primary culture. Here we can appreciate how in this specific condition in which basal cell were cultured for extension, we lose some of the heterogeneity that we see from freshly isolated basal cell. And this is an important consideration when we want to expand, for example, for a specific subset, because it indicates we really need to focus on optimizing the culturing condition tailored to the specific subset that we want to expand. And this methodology was, of course, also very useful to specific subset of basal cell and analyze them in vitro. In this case, this is a comparison of canonical basal cell with the secretory prime basal cell. And we can see when we clonally expand them as an organoid, they start to differentiate more quickly and start to produce specific mutants. Another thing that we were able to use our atlas for was to envision models that can be used for in vivo analysis of the transitions that we are observing in vitro in our in silico analysis. For example, we were very interested in pursuing the analysis of transition between basal cell subsets and secretory cell, because this is obviously a phenotype that is enhanced in cystic fibrosis. And so what we did is to produce a mouse model in which we were able to lineage tag a specific subset of secretory cell that we know being in relationship with transitional basal cell from our in silico analysis. And here is description of the model in which SGB1A1 and SGB3A2 are used to drive expression of CRER and 3ER. They ultimately will label the different subset of secretory cell in green through GFP and red through the tomato. Andrew Beppo in the lab is specifically using this tool to analyze transitions between subset of basal cell and subset of secretory cell in the context of lung injury in the mouse. And is using this model in the context of H1N1 virus infection. Here is an example, proof of concept that the model works. You can see how you can nicely label different subset of basal cell, or secretory cell, in green and red. And here is the experimental model that is using with H1N1 virus. And with just keeping really a long story short, the take-home message here is that performing this type of specific injury, it was able to observe that not only we can observe a transition of secretory prime basal cell to secretory cells, but also in this specific context, a subset of secretory cell are able to go back and transition to a basal cell phenotype. And this is an important consideration when we want to think about which cell could be potentially used for therapy, because it indicates that in a specific condition of injury and insult of the lung, some secretory cell may as well be considered for their ability to revert to a basal cell phenotype. So today I've shown you how the atlas that we have built is a very important tool that can be used for the cystic fibrosis community. I show you a couple of examples how it can be used to devise strategies for fractionation of specific cell subtype that we classified in our analysis. And it can also be used to envision in vivo model that can be adopted to really further describe the transitional state that we are able to identify in our analysis. Overall, this work has some limitation that we need to acknowledge for the fact that it was using really normal healthy airways in comparison to end-stage cystic fibrosis disease. Therefore, we miss all the information related to early events that happen during the development of the disease. For this reason, we are coupling this analysis that we already performed with a specific model, ferret. There is an animal model that developed cystic fibrosis in a similar way to what happened in humans. In particular, in collaboration with the University of Iowa, John Engelhardt lab, we are using this ferret model in which a gating mutation was introduced, the G551B. It's also a mutation that is present in people with Cftr, with cystic fibrosis, sorry. And by virtue of adding the X770 or removing the drug, we are able to keep the animal relatively healthy or trigger the onset of early step of cystic fibrosis. And this is a very powerful tool because now we have the capacity of study early event and exacerbation of the disease. It also have the very useful use of analyzing what are the characteristics of cystic fibrosis at different steps of the development of the disease that can be possibly reverted. And this is going to be used as a very powerful tool really to see what are the phenotypes associated with cystic fibrosis that are not revertible from the one that may be revertible. So this will be a very useful information that we can gather from the analysis of this transcriptomic analysis that we are performing and comparing that with what we already did in the human context. I'd like to thank everyone for listening, and thanks all the members of the lab, the external collaborators, as well as the funding from the Cystic Fibrosis Foundation that really allow this project to become a reality, and NHLBI.
Good morning. First of all, I would like to thank Margarida and Barry for the opportunity to present our work in this exciting workshop. My name is Hugo Botelho, and I am a junior researcher at Margarida's lab at the Biosystems and Integrative Science Institute from the University of Lisboa. And in my talk, I will tell you how we are using functional genomics to identify novel therapeutic targets for cystic fibrosis and how this work can push current CF biomedical research forward. I have no relationship to disclose regarding this presentation. The panorama of pharmacological management of cystic fibrosis has dramatically changed over the last few years with the introduction of Cftr targeting modulators, all acting at the Cftr mutation-specific level. For the first time, we were able to directly address the basic molecular defect originating CF for improved quality of life. Even though ongoing preclinical research and development of novel Cftr modulators is expected to improve the genetic coverage within the CF population, there is still room for improvement, especially regarding rare Cftr mutations. For most mutations, pharmacotherapy is based on the combination of character and potentiating molecules. The need for combination arises from the diversity of molecular defects imparted on the Cftr molecule by mutations, which can be grouped into 7 mutation classes, and from the possibility of a given mutation to give rise to multiple simultaneous defects. The foremost example is the most common mutation, F508del, which concurrently integrates classes 2, 3, and 6 due to a combination of protein misfolding, deficient plasma membrane traffic, gating defects, and reduced plasma membrane stability. Globally, release from the endoplasmic reticulum is the major obstacle for F508del Cftr rescue. The mechanisms responsible for ER retention of F508del Cftr are still not completely elucidated, but there exist at least 4 sequential checkpoints which oversee the folding of the Cftr molecule, starting from the moment the polypeptide is translated. In order, they are the co-translational surveillance by members of the HSP70 chaperone family, association with the ER-resident chaperone calnexin, retention of Cftr molecules exposing 4 arginine frame tripeptides, also called AFTs or RXR motifs, and the binding to the COP2 machinery for ER2-Golgi transport via a DAD diacetyl code. The involvement of uncharacterized genes into the ER-quality control checkpoints can be deconvoluted by observing their effect on Cftr mutants with differential susceptibility to each checkpoint, either with second-site mutations which conformationally correct F508del, mutations which affect the 4 AFTs, or which mutate the 2 aspartate residues in the diacetyl code required for ER export. Systems biology techniques are invaluable for the discovery of novel Cftr-regulated genes. They allow not only the identification of new therapeutic target genes, but also the biological pathways or hubs potentially relevant in Cftr rescue. Eventually, this preclinical biomedical research is expected to benefit individuals with CF by providing novel and more effective therapeutic solutions. Our approach to identify global F508del Cftr traffic regulators was based on a Cftr traffic reporter system we described a few years ago. The Cftr traffic reporter is a double-tagged Cftr molecule containing an M-cherry fusion at the N-terminus and a flag tag inserted into Cftr's 4th extracellular loop. The traffic reporter is expressed by CFB cell lines under the control of an inducible TETON promoter. Addition of doxycycline to the culture medium triggers expression of the construct, whose total expression can be measured with a fluorescence microscope by the M-cherry fluorescence intensity. If the construct traffics to the plasma membrane, the flag tag becomes exposed to the extracellular space, and the plasma membrane pool can be quantified by immunostaining the flag tag in unpolymerized cells, using, for instance, a secondary antibody conjugated with Allele 6 or 7. Then, traffic efficiency can be calculated by the ratio of plasma membrane versus total Cftr. These fluorescence micrographs illustrate the ability of the reporter cell lines to translate the characteristic cellular distributions of wild-type and F508del Cftr. Whereas wild-type Cftr accumulates into CFE cells as seen through the M-cherry signal and is detected homogeneously covering the cell surface here, the F508del Cftr steady-state concentration is much lower, and only residual plasma membrane staining is detected. In this work, we hypothesized that there exist unknown cellular factors which retain F508del Cftr in the endoplasmic reticulum, which can be inhibited to promote plasma membrane traffic. These genes are therefore potential novel drug targets for CF, whose activity may be further enhanced through synergy with current Cftr modulators. We tested this hypothesis with a functional genomics screening campaign. For the screening assay, multi-well microscopy assay plates were coated with a mixture of sRNA intersection reagent, the so-called reverse transfection protocol. Cell seeding triggers transfection and knockdown of one gene in each well. After allowing 24 hours for the target gene to be depleted, CFTR expression is induced with doxycycline, and 48 additional hours are allowed for CFTR synthesis and traffic. Then, extracellular FLEC tags are immunostained in unpermeabilized cells, and epifluorescence imaging is performed. Finally, a high-content image analysis pipeline is used to segment cells, excluding unrepresentative ones, and fluorescence quantification is performed on each approved cell. In this talk, I will show the quantification of plasma membrane CFTR expressed as Z-score. We use this assay to screen Ambion's extended druggable genome siRNA library, which targets 9,128 human genes with 3 distinct siRNAs per gene. After filtering siRNAs targeting non-protein coding genes or having off-targets and excluding also unusable assay plates, we quantified plasma membrane traffic following the knockdown of 8,592 genes. We defined the Z-score of 2 as our hit threshold. This resulted in the identification of 228 siRNAs targeting 227 genes as significantly enhancing FA-Del CFTR traffic. The potential relevance of enhancer hits can be better appreciated when comparing the plasma membrane quantification with that produced by incubating control cells with VX809 at the 3 micromolar concentration and observing the high plasma membrane score, as one can see in this inset plot here. To confirm the results from the primary screen, hits were rescreened with the same bioassay using distinct siRNA molecules, 2 per gene whenever possible. Therefore, a secondary siRNA library was assembled comprising 450 siRNA molecules targeting 225 genes. These experiments confirmed 35 gene knockdowns and 70 siRNAs as significantly enhancing FA-Del plasma membrane traffic, a confirmation rate of 15%, again with a magnitude larger than VX809 as one can see. This suggests that the 35 genes are ER retention factors which can be downregulated to restore FA-Del CFTR traffic. Here is a small set of microscopy images from cells treated with hit siRNAs. In the left, we have control cells treated with the non-targeting NEG1 siRNA. Knocking down the CFTR gene significantly reduces end-cherry fluorescence, indicating high transfection efficiency. Then, treatment with VX809 or a selection of hit siRNAs significantly enhances the levels of plasma membrane CFTR, as also quantified below. I am not able to reveal the identities of hit genes, but I will follow some of them using coded names, including these kin genes, which correspond to kinases. We measured the expression of the 35 traffic-regulated genes in human native lung tissue and CFE cells by RT-PCR. 89% of hit genes are expressed in lung tissue, including all 3 kinase genes, kin1, kin2, and kin3, demonstrating the physiological relevance of the hits. We then performed an orthogonal biochemical validation of the 35 gene knockdowns. We transfected CFE cells expressing unmodified FA-Del CFTR with siRNAs targeting each gene, the same 2 siRNAs which were part of the secondary siRNA library. We then detected CFTR using Western blot and performed the cytometry quantification of BENC, which represents the fully processed population characteristic of the plasma membrane. We observed that almost all knockdowns generated robust increases in plasma membrane CFTR when compared with the non-targeting siRNA control represented by the dashed line. Interestingly, some of the largest rescues were observed when knocking down the 3 kinases, kin1, kin2, and kin3. Nevertheless, we were somehow surprised by the remarkable rescue performance of the 35 top scoring genes, and wondered whether relevant rescue could also occur with some of the suboptimally scoring siRNAs. So we performed additional Western blot experiments, and were happy to observe that the knockdown of an additional 18 genes, which were just below the hit threshold in the confirmation screen, also generated robust BENC rescue. Some of the highest rescue levels were obtained when knocking down 2 additional kinases, kin4 and kin5. At this point, we considered that the data we had gathered so far and decided to select the 5 kinase genes, kin1, 2, 3, 4, and 5, whose knockdown yielded FA-Del Cftr rescue for further studies. Even though kin4 and kin5 did not meet the hit threshold on the confirmation screen, we still considered that the Western blot data was robust enough to justify the decision. The selection of a subset of 5 kinase genes was evidently based on the amenability to small molecule inhibition. To ascertain how rescue efficiency via the knockdown of the 5 hit kinases compares with Cftr clinical characters, and if there are additive effects, we knocked down each of the 5 kinase genes in CFBE cells overexpressing FA-Del Cftr. Cells were tested as controls versus incubation with VX809, VX661, and the VX661 plus 445 combination. We observed that several corrector treatments are additive to kinase knockdowns. In most cases, additivity is positive, but when kin4 is knocked down in the presence of VX445, we observe decreased rescue. These data suggest that some hit kinases are potential novel FA-Del drug targets, acting on traffic by distinct mechanisms of action from approved corrector drugs. To test whether the kinase knockdowns also restore FA-Del Cftr function, we performed patch clapping experiments. Functional rescue was observed when knocking down kin1 through kin4, but not kin5. Overall, the most effective knockdowns were those of kin1 and kin4, not only for the magnitude of functional rescue, but also because of the additivity with corrector VX809. At this point, we aimed at the mechanistic description of the connection between the hit kinase knockdowns and the FA-Del Cftr rescue, trying to identify the biological pathways they may act on, or if they target any of the quality control checkpoints. To ascertain the overall pathways targeted by the hit kinases, we compared our primary and confirmed screen hits with published datasets of CF or CFTR regulated genes. We considered the FA-Del Cftr interactomes reported in the papers by Riley, Kanatu, and Pankov, the secretion machinery from Simpson et al., genes regulating the activity of FA-Del Cftr or the epithelial sodium channel ENaC, the papers from Tomati et al. And Almas et al., as well as CF lung disease modifier genes from Dung et al. In this plot, each line corresponds to an individual sRNA or gene, possibly colored according to the screening scores. We started by finding all genes which occur in more than one study and connecting them with lines in this plot. We noticed that our hit genes were rarely reported in other studies. We then looked at the 5 hit kinases which we had selected, kin1 through kin5. Surprisingly, there was only one instance where one of these kinases was part of any other dataset. This was kin4, which has been reported as an ENaC regulator. Overall, this analysis suggests that FA-Del Cftr traffic screen hits are a novel category of Cftr regulators. This claim is also supported by the fact that the 5 hit kinases were not hits in other sRNA screens, which assessed FA-Del Cftr rescue when screening druggable genes or the kinome. We are referring to these 2018 papers from the lab of Nicolette Pedemonte and Marcel Boucher. To determine whether the 5 kinases interfere with the quality control checkpoints, we performed knockdowns in CFBE cells expressing second site FA-Del reverted mutations, including combination with VX661 or VX661 plus 445. I don't have time to describe these results in detail, but the major conclusion is that several hit kinases have a differential contribution to quality control mutants and folding correctors. For several knockdowns, one can observe additivity with the G550E and R1070W mutants, which alleviate the first quality control checkpoint, as well as the 4RK mutant, which partially circumvents the third checkpoint. There is also additivity with some correctors, even though VX445 is deleterious in some situations. We did not find any effects with the DDAA code mutant affecting the fourth quality control checkpoint, and we are currently mapping our data on kinase knockdowns in the presence of FA-Del Cftr revertants and correctors to propose a model on the underlying mechanistics, which we briefly outline here. This classification task is still ongoing, though. In the meanwhile, we were particularly excited on F-avoid-del Cftr rescue through Kin4. The Kin4 knockdown had the best performance in rescuing F-avoid-del Cftr in most assays, notably in patch clamp. Additionally, the additivity to VX661, possible overlap with the pathway targeted by VX445, and relation with all ER quality control checkpoints are valuable tools for mechanistic analysis. But perhaps more importantly, through literature search, we uncovered the biological pathway which could account for the rescue phenotype and for which there are inhibitory drugs which may be tested for repurposing. Finally, a selective Kin4 inhibitor with nanomolar IC50 was recently reported. We therefore synthesized the Kin4 inhibitor and performed a dose-response experiment, assessing whether Kin4 inhibition could rescue F-avoid-del Cftr. These experiments showed that, indeed, low micromolar concentrations of the Kin4 inhibitor rescued F-avoid-del Cftr band C. Rescue was not at the level of VX661, but was nevertheless significantly increased versus the control, suggesting that Kin4 may be a novel potential therapeutic target for CF. I should stress that no siRNAs were used in this experiment. In summary, I've told you how we have performed the high-content siRNA-based microscopy screen, targeting about 9,000 human genes, and identified 225 potential regulators of F-avoid-del Cftr traffic. Through a combination of hit validation, human lung expression, and comparative analysis with other datasets conveying global results, 5 kinase hits were selected as novel F-avoid-del Cftr traffic regulators. Some hit kinases are potential novel F-avoid-del Cftr drug targets, acting on traffic by distinct mechanisms of action from approved corrected drugs. Finally, we propose Kin4 as a novel CF therapeutic target, which can be inhibited with a small molecule to achieve F-avoid-del Cftr traffic rescue. I will finish by acknowledging my lab, headed by Margarida Amaral, and the people who contributed to this work: Miquel Lopes-Pacheco, Madalena Pinto e Silva, Luca Clark, Cristina Moiteiro and Vasco Cftr for the inhibitor synthesis, the Advanced Light Microscopy Facility at Embol headed by Raina Peppercock, which was instrumental in the beginning of the project, Carl Kunzmann's lab for the patch clamping infrastructure, our local high-throughput screening facility, and funding from our national funding agent, FCT, the European Union and CFF. And finally, even though this is the best I can do in terms of a group picture in these troubled times, I want to thank again the contributions of those directly involved in the work, and also to all of you for your attention. Thank you very much.
Hello, everyone. We have a couple of questions already from the audience, and I'll be reading these questions. And the speakers will be addressing these questions directly. So the first question, this is for Brian, is from Patrick Harrison. So Brian, do you expect the same splicing or mid-splicing pattern of superexon in cells derived from the nasal cells?
Great question. The quick answer is we don't know. But especially as one tries to develop a methodology that can work, as you've indicated, either in nasal epithelial cells or, let's say, intestinal cells or pancreatic cells in addition to the lung, this is going to be a really important issue. And there's a lot of evidence already in the literature that splicing patterns can vary significantly from tissue to tissue or cell type to cell type. So I think this is something we're going to have to follow up as I think doing iPSCs and then differentiating them into different cell types is probably one way to address that directly.
Okay, we have another question from also directed to Brian, and the question is from Janet Allen. So Janet found your talk very interesting, and this is for the experts. Inserting the cDNA 9 to 27 into intron 8, what is the frequency of insertion into both alleles compared to one allele? Is there a subsequent effect on cell behavior if both or one allele are inserted?
It's a great question. In principle, if one has a target integration rate of, let's say, 50% per allele, then in general, just from probability, one would expect that 1/4 of the cells would have no target integration, 1/4 would have homozygous integration, and then 50% would have integration at either one allele or the other. But what's been found in editing work over the last several years is that in fact, the frequency of homozygous targeting, that is targeting at both alleles, is higher than you would expect based upon that simple probability. It seems that cells that are, the suggestion is at least that cells that are amenable for target integration, that they can in fact incorporate it experience editing at both alleles more frequently than you would expect based upon that frequency. So in fact, in the data that I was showing in the splicing experiment, where we actually looked at single cell derived clones, and we wanted to pull out those that were homozygous targeting for the purpose of studying carefully this alternative splicing that Patrick addressed in his question, we found that in fact that an unexpectedly high frequency of the clones that we looked at were in fact homozygous targeting. So it was not hard at all coming up with the 4 clones reported shown in that figure. So we don't really have a careful determination of that. I think probably ddPCR is going to be the best way to address that on a single cell basis. With respect to whether there is a particular proliferative advantage or the like on homozygous targeted cells, we don't know. It's a great question, and maybe that helps explain why our frequency was higher than expected, although I doubt it. But I don't have a good answer to your second question, which is functional consequences in terms of proliferation.
Thank you, Brian. So I have now myself a question to Gianni. So we have used BCI cells from Ron Crystal in Cornell, and we actually have modified them into F508del expressing cells. But altogether, still for the wild type cells, we have recently shown, still data submitted for publication, that basal cells start by differentiating into suprabasal population that can differentiate into pulmonary ionocytes and club cells. And then club cells are an intermediate population that can still differentiate into ciliated and goblet cells. Do you also have, do your data support this as well?
Yes, so we do see that transition as well from the in vivo data. And I will actually say that is probably the canonical transition that happened in the normal lung in the physiological condition. And then the other transition that we describe, in which you have direct differentiation from basal to ciliated, I would consider it more of a pathological onset that happening in some type of injury like the one that we see in CF. I guess the other comment I can add is we also try to do some in vitro follow-up experiment. And we find that depending on the type of media that we use, so culture condition, we may push toward one way or another. And the basal to secretory differentiation is probably the easiest one to achieve. And then I guess the question regarding ionocyte, at least from the transcriptomic data, is difficult to infer just because the population of ionocyte is so small. There is no easy to infer transitional state just by the transcriptomic data. And then that you need to do experiment like the one that you described in vitro to be able to address that part.
Thank you so much. We have now a question to Hugo from Lars Plate. Lars actually thanks you for the great talk, and his comment is that it is curious that most kinases were not identified in prior interaction datasets. Do they not directly phosphorylate Cftr, and does that indicate an indirect mechanism or action to restore trafficking?
Thank you, Lars, for the question. Yes, indeed. So as far as we have looked into the subject, none of these kinases seem to directly interact with Cftr, and as far as others have reported in the literature. So we are hypothesizing that their effect on Cftr traffic is indeed an indirect effect, and we're not sure exactly what are the mediators in this indirect effect. This was part of the reason why we started looking into ER quality control checkpoints to start to get an idea on what could be some of the connecting steps. But it's not clear for us at the moment. We are pretty sure that it's an indirect effect, but we are still trying to come up with a holistic view of how this can turn out on how knocking down this kinase can rescue Cftr traffic specifically for F5 breakdown.
Yeah, if I may add also something, I guess my impression, although these studies are global studies, they are always partial. So no study is completely global by identifying all interactors, and so despite that they are global, they are partial. Another question for Hugo from Barry. My co-moderator who's at the back office. So Hugo, it's a very nice presentation. Barry says he may have missed in your presentation, but what level of redundancy exists between the hits identified in your screen? And this may shed light on the complexity of the pathway involved in Cftr trafficking.
Yes, thanks very much for the question. You haven't missed the data, because indeed we have experimental data on that, but I didn't show it in the presentation. So we actually tried combining the knockdown of multiple kinases at once to get an idea of whether they could be redundant or not, and the picture is quite complex. So some of them are redundant, some are not. In other words, we can see additional effects when knocking down two kinases at once, but this does not work for all combinations, and because the picture is somehow complex, I didn't include the data on the presentation. But yes, so some of them are redundant and some of them are not.
Thank you. So another question for Hugo from Janet Allen. So interesting, thank you. Checkpoints for misfolded proteins are like safety mechanisms for cells. And how specific do you think the kinases actually targets kinase targets identified other misfolded proteins? Is this a problem for new therapeutics? It's about specificity.
Yes, thanks, Janet. So, just to start answering, our goal in using S07 reverting mutants was to get an idea on the mechanistic, so that we could assign some of the hits to one of these checkpoints. So it's not that we are looking into modulating some of these checkpoints for potential biomedical applications. It's just for us to get data on how these knockdowns could be translated into CFTR arrest, because indeed they are safety mechanisms for cells. We so far haven't explored specificity, so we haven't tested whether whether knocking down these kinases affects other proteins other than CFTR. But we do have data which I haven't shown, that they are somehow specific for F508del, so they do not rescue other class 2 mutations. So there is some specificity for F508del, but we are yet to test on non-CFTR targets.
Thank you. So another question for Hugo from John Hunt. Hi, John. Are there any interpretable themes in the data you showed quickly comparing drug responses when modulating different pathways? Is there a clear division between pathways modulating lumacaftor, tezacaftor versus elexacaftor?
Yeah, so we still don't have a clear idea on the pathway. So some of the analysis we have been doing has been centered around trying to understand the additivity with the tezacaftor and the elexacaftor, so because we see that for most of the knockdown we get additivity with tezacaftor, but the additivity is lost when we add elexacaftor to the mix. Our hypothesis is that some of these kinases are regulating probably this one of the elexacaftor binding sites. So it's not. So I believe that it's not clear to the community where elexacaftor might be binding to CFTR. There is a preprint from several groups, including Isabel Calboz, where they propose two binding sites for elexacaftor, one in NBD1 and another one on the membrane spanning domain 1, and probably because this MSD1 binding site seems to be specific for elexacaftor, we might be somehow targeting that site with some indirect interactor of these kinases, but we don't have any hard data on that. It's just our working hypothesis.
Thank you very much. I think there are no more questions. Okay, so I think the session is now over. I just left to thank all the speakers, thank all the participants, and of course my co-chair, who you cannot see at the moment, but Barry Strip has been a major help at the back office dealing with the questions. And thank you very much. Hope to see you at the other sessions of NACFC.